Hi everyone, Queen Nerdling here. Today we're going to be talking about translation. So in this video, we're going to be examining how the messenger RNA transcript is translated into a protein. So during translation, we convert messenger RNA sequences and the information that they hold into these polypeptide sequences. And this process occurs at the organelle called the ribosome. So the genetic code is interpreted as a series of 3 nucleotides. These 3 nucleotides come together and they form what we call a codon. So 3 nucleotides are coded for by a codon on that messenger RNA strand. There are 64 possible codons that code for 20 amino acids, along with one very special start codon, AUG, and 3 different types of stop codons, UAA, UAG, and UGA. And so as you see here, we have our messenger RNA strand. And for example, the very first codon, AUG, is going to code for the amino acid methionine. And this is referred to as the start codon. Now after that, every 3 nucleotide bases forms a codon, and each of those codons is going to code for an amino acid. Now at the very end of this protein or polypeptide chain, we're going to have a stop codon. In this sense, this one is UAA, and it signals for that protein synthesis process to stop. So next up, we're going to look at these 2 different codon charts. You are probably more familiar with one of these charts over the other. However, I suggest you make sure you know how to use both of them, just in case you're only given one form of this on an exam. Now, the easiest one, in my opinion, to use is this circular codon chart. So if we're going to figure out what codons does AUG code for, or what amino acid does AUG code for, we'd start here. So we'd start in the center of this. We start with the first letter, which is A. Then we go to the second letter, which is U. Then we go to the third letter, which is G. So if I went A, U, then G, we know that it codes for methionine, which is, surprise, surprise, the start codon. Now looking at our different types of stop codons, I'm going to go ahead and use the second type of codon chart. So if we're going with UAA, we have to look over here. So we're going to look at the first base in the codon, and we're going to use this first column here. So the first letter is U. So this is the box that we're going to start with. The second letter is A. So we now have U, and then we have A. Our third letter, which is represented over here in this sequence, is A as well. So we have U, then we go over to our A, and then we have our second A right here. So we basically cross it, and it codes for our stop codon. Now, as you can see, I feel like this one is slightly more complicated to use than our circular codon chart. But like I said earlier, it's a good idea to make sure you know how to use both of these in case you're only given one on an exam. So ribosomes are the site of protein synthesis, and ribosomes are composed of ribosomal RNA as well as protein. These are the only non-membrane bound organelle, and all cells have ribosomes, whether we're talking about prokaryotic cells or eukaryotic cells. These ribosomes have 3 different sites that each of the transfer RNA molecules moves through during the process of translation. We have the A site, and this is called the aminoacyl site. This is where transfer RNA is going to attach. We then have the P site right up here, and this is the peptidyl site. This is when transfer RNA holding that polypeptide, or I should say the amino acid, is going to form a peptide bond between the transfer RNA holding the amino acid next to it. So these 2 amino acids right here are going to form a peptide bond. Once that peptide bond forms, this transfer RNA will then move into the E site, which is the exit site. It will have dropped off its amino acid, and it's empty. So this peptide bond right here is stronger than the hydrogen bonds that are going to be holding the codon and the anticodon together. So we have our A, or aminosyl site, we have our P, peptidyl site, and then we have our E for exiting site. And that transfer RNA follows that process. It attaches, forms peptide bonds, and then it exits. So just like in the process of transcription, translation too has initiation, elongation, and termination. So messenger RNA interacts with the large subunit of the ribosome to begin the translation process or to initiate the process. This occurs at that start codon AUG. And remember that AUG is coding for the amino acid of methionine. So this is closest to the 5' end of the messenger RNA strand. The transfer RNA anticodon that's containing that start codon of methionine is going to bind at that P polypeptide site. The small ribosomal subunit is then going to attach, forming the initiation complex. So again, we have our small complex right here. Our messenger RNA is going to come in here with our start codon methionine. Then we're going to have the transfer RNA anticodon that has that methionine bind right here at our P site, the polypeptide site. Then this small ribosomal subunit is going to attach to the larger subunit, and this forms the initiation complex. Once this occurs, more of our transfer RNAs can come in and drop off their amino acid, which will eventually start to form that polypeptide chain. And that brings us to the elongation process. Those subsequent amino acids are brought to the ribosome with transfer RNAs. So as you see here, we have our elongation process. We have a transfer RNA that's going to come in. It drops off its amino acid. This amino acid is going to form a peptide bond with the amino acid next to it. Once this peptide bond is formed, the transfer RNA can break the hydrogen bonds that are holding together the codon from the messenger RNA to the anticodon that the transfer RNA is carrying. Now the transfer RNA leaves empty-handed because it drops off that amino acid. And again, that peptide bond is stronger than the hydrogen bond. So this will then go out through the E site or the exit site, and this transfer RNA can go on to pick up another amino acid. Next up, we have another transfer RNA that will come to the attachment site. It will move on to the P site where it will form another peptide bond, and the same process will occur until we reach a stop codon. And that is the beginning of our termination. So this elongation process occurs until we reach that stop codon. This is going to trigger the release of the polypeptide, and the ribosome will then disassemble. And so if you recall, each of these is an amino acid, and those amino acids are being held together through a peptide bond. Many amino acids linked together is called a polypeptide chain. So again, once we get to that stop codon, it's going to signal the release of that chain, and the ribosome is going to disassemble. So what is the difference between prokaryotic and eukaryotic translation? Well, as you recall, prokaryotic cells lack a nucleus. So the transcription and translation process are coupled and occur simultaneously. Whereas in eukaryotic cells, proteins can be made on free ribosomes, which are just floating around in the cytoplasm. Now, when they're synthesized at these free ribosomes in the cytoplasm, those proteins are typically used in that immediate area. In eukaryotic cells, because they have those membrane-bound organelles that serve different functions within the cell, we also have rough endoplasmic reticulum. Now, the rough endoplasmic reticulum is studded with ribosomes, and those ribosomes are going through the process of protein synthesis. Now, proteins that are synthesized on the rough endoplasmic reticulum typically will get transferred to the Golgi apparatus. That Golgi apparatus will then package those proteins into vesicles and ship them out where they need to go. Many times they'll be secreted from the cell, or they'll be incorporated into the plasma membrane. So to kind of do a big recap of the entire process of protein synthesis in a eukaryotic cell, we're going to first start here in the nucleus. We're in the nucleus, and the first portion is going to be transcription. That's where we are going to take the template DNA strand, and we're going to make a complementary messenger RNA strand. The enzyme RNA polymerase is responsible for breaking those bonds, as well as synthesizing that messenger RNA strand by adding the complementary bases, so our nucleotide bases. Now, once that messenger RNA strand is synthesized, we call that the pre-messenger RNA strand because it contains both the introns and the exons. And it has to be processed in order to be able to leave or exit the nucleus. And we call this RNA processing. During RNA processing, 3 different things occur. We're going to add a poly A tail, which is a series of adenines, thousands of them, added on to the 3' end of the messenger RNA. We're also going to add a 5' G cap. And so we're adding a modified guanine molecule to the 5' end. Those 2 components act as almost 2 different types of keys that allow that messenger RNA to leave or exit the nucleus. It prevents them from being damaged by different enzymes in the cytoplasm, and it also acts as a key for the nucleus to actually allow them to exit. But before that strand can exit, we need to take out the introns. Introns are sequences of DNA that don't code for a protein, and we need to have a way of removing them so they don't get exported outside of the nucleus. And we do this through a process called splicing. Splicing occurs at a spliceosome. So a spliceosome will basically remove the introns. It will splice together the exons through using ligase. And then we have our functional messenger RNA strand. The introns stay inside of the nucleus, and the exons get to exit. So the messenger RNA strand has been processed. We have that poly A tail, and we have that 5' G cap, as well as only the exons. That exits the nucleus, and then it is able to be synthesized. So this is going to find a ribosome. That ribosome is going to have the initiation complex that forms. So we're going to bring in methionine. When methionine is brought in, it basically causes that ribosome to create the initiation complex. Once that initiation complex has been created, then transfer RNA molecules can bring in their amino acids. Those transfer RNA molecules bring in the amino acid as well as the anticodon. Anticodons bind with the codons. Once this occurs, this transfer RNA is going to move to the P site. So it starts off at the A or attachment site, moves to the P site. I always like to say P is for peptide. This is where we're going to start forming our peptide bonds. So between each amino acid, we're going to have a peptide bond. Now these peptide bonds are much stronger than the hydrogen bonds, which are connecting the codon and the anticodon. Once these peptide bonds are formed between 2 amino acids, the transfer RNA is going to break those hydrogen bonds, and it's going to be released. This can now go on to pick up another amino acid. Eventually, one of those transfer RNAs is going to carry with it a stop codon. Once that is read, this process will stop, and that amino acid sequence, which is now formed, a polypeptide chain, will be released. Then it will undergo a process of folding so it can turn into a fully functional protein. So Speaking of fully functional proteins and polypeptide chains, I'm bringing back the tangle beads. So I've used these in biochemistry to talk about protein folding and the different conformations of a protein. So basically what we've described in the process of translation today is up through the point of forming that polypeptide chain, where each one of these individual tangle beads would be an amino acid. Each of these amino acids had been coded for by a codon. And so those amino acids are linked together through our polypeptide bonds. Now this would be the primary structure of a protein. After that, the protein is going to start folding, and we're going to get two different structures. Now we can have an alpha helices that forms, and then we get hydrogen bonding that forms between each of the rings. Or another type of secondary structure is that beta pleated sheet. And again, we'll have hydrogen bonds between the beta pleated sheets. Now, we can move on to the tertiary structure of a protein, which is a combination of beta pleated sheets as well as our alpha helices. We might have more than one alpha helices or more than one beta pleated sheet. Now, in that secondary structure, we're forming hydrogen bonds. In the tertiary structure, we're starting to get stronger covalent bonds, such as disulfide bridges, as well as ionic bonds. Now, we might stop here for a fully functional protein, or we could go into a quaternary structure where two or more of these tertiary structures or subunits combine together to form a quaternary structure and become a fully functional protein. Well, I hope that was helpful and that you've learned about the process of translation. This is the Queen Nerdling signing off for today. For more videos like this one, you can go to www.nerdlingscience.com.